Many of you may have questions about genetically modified organisms, what that means, what that really entails. To completely understand those ideas, we need to first explore this idea of gene expression. So during this exciting week, what we're going to look at is this connection between DNA and genetics. What we're going to first look at is this idea of gene transcription. So overall in gene expression, we are trying to take the information stored in DNA and use that information to manufacture a proper protein. Proteins, remember, do all these functions inside of the cell. To get to that point of making the protein, we need to start with transcription. And this is simply taking the DNA, that information stored in DNA, and using it to transcribe a molecule of RNA. And remember, DNA and RNA have a lot of similarities. They're both made up of nucleotides. So when we take this DNA and manufacture an appropriate strand of RNA, we call it transcribing or transcription because they are very similar molecules. It's basically making a copy. This needs to occur because RNA acts as like this middleman. This messenger RNA acts as a middleman and allows the information stored in DNA to be able to leave the nucleus. Remember, we have over 2 meters of DNA stored inside of that nucleus. That's a lot to try to move outside of the nucleus. So instead of moving all of the DNA, we take just the information for one gene that will code for one protein and just manufacture a shorter segment of RNA that is actually able to leave the nucleus. So as we look at transcription, we have to start understanding that this is taking that DNA code, those sequence of A's, T's, C's, and G's, and copying them into a sequence of RNA. And this specific sequence of RNA is called messenger RNA, or mRNA. And it's taking those A's, T's, C's, and G's of DNA and making a sequence of A's, U's, C's, and G's in RNA. This all occurs inside of the nucleus. To facilitate this building of messenger RNA, an enzyme is required. And the specific enzyme is called RNA polymerase. Remember, ASE always refers to these enzymes. And remember, a polymer is a long chain. So this is an enzyme that makes a long chain of RNA. It's called RNA polymerase. This enzyme functions inside of the nucleus. It has a couple important roles. One of the roles that needs to happen, one of the jobs that needs to be performed is taking that double helix of DNA and breaking it open, exposing those nucleotides. RNA polymerase is able to open up that double helix and then incorporate the proper RNA nucleotides to manufacture that chain of messenger RNA. So let's take a little bit of a closer look inside of transcription. So what you see in this gray background, that is the RNA polymerase enzyme. You should see extending to the left and the right in the blue molecule, that's DNA. And to the left and the right, the extreme ends, it's a double helix. There's base pairing holding it together. But where that RNA polymerase is, the hydrogen bonds holding those nucleotides together have been broken. This is called the transcription bubble, and it allows RNA nucleotides to be incorporated. And you should see the same type of base pairing occurring. A G nucleotide in DNA would base pair with a C nucleotide in DNA. Same thing happens in DNA to RNA. The only difference is if you have an A nucleotide in DNA, it would base pair to a thymine or a T nucleotide in DNA. RNA doesn't have a thymine. Instead, it has a nucleotide called uracil. So you see an A base pairing with a U instead of a T in RNA. So it's a subtle difference, but it still maintains and holds equivalent information. Another thing you should notice in here is it's a little bit different than DNA replication. During replication, both strands of the DNA are copied. In transcription, only one of the two strands of DNA is actually copied into a strand of mRNA. The strand that is copied is called the template strand, or the template strand of DNA, and that's used to manufacture the appropriate messenger RNA molecule. And that becomes extremely important when you're trying to manufacture a very specific protein. Only one of the strands of DNA can be used. After this is all complete, so you initiate transcription at something called a promoter site, and the RNA polymerase moves along the strand of DNA to make the complete transcript. That's the final messenger RNA product. That messenger RNA can now leave the nucleus and attach to a ribosome. And at the ribosome is where that next process occurs, where that process called translation. So we should start to have this understanding of the steps that are occurring in transcription. So we're going to look at drawing it to help understand what's going on. So as we look inside of a cell, so this is a cell, could be your cell, my cell. We look inside and we see a nucleus. Within that nucleus, you're going to have a strand of DNA. That strand of DNA is going to be too large to be able to leave the nucleus and attach to a ribosome on the outside. So this is our ribosome. So we need the middleman. Within that string of nucleotides that make up DNA is something that codes for a specific protein called a gene. This is just a sequence of nucleotides coding for one protein. That gene will be transcribed into a single chain of messenger RNA. So that's our final product. This messenger RNA can then leave the nucleus and attach to the ribosome for translation. So let's take a closer look. So I'm going to unwind the DNA to make it a little bit easier. So we have this unwound chain of DNA. Let's even put some letters in there. So let's say we have A, T, C, C, G, and A. The corresponding strand of DNA would be T, A, G, G, C, and T. Those are being held together by hydrogen bonds. If we want to make a chain of RNA, we have to break those hydrogen bonds. So the enzyme, RNA polymerase, can come in and bind to that specific sequence of DNA called the promoter sequence. We won't talk more about that. That can come later when we talk about regulation of gene expression. So right now we're just looking at RNA polymerase binding to this promoter site. We need to break the hydrogen bonds holding the DNA together. So we can go down here and say they've been broken. So we broke the hydrogen bonds. We still have the same sequence, A, T, C, C, G, A. And up here we have T, A, G, G, C, T. And remember, not both strands are going to be transcribed, only the template strand. So we'll call this bottom one the template strand of DNA. We still have the enzyme RNA polymerase attached to the DNA, and it's going to now transcribe the appropriate strand of messenger RNA. That messenger RNA is going to be transcribed in the 5' to 3' direction, meaning this strand of DNA is in the 3' to 5' direction. Remember, they're always going in the opposite orientation. So we transcribe this strand of DNA. Base pairing rules apply. A's, instead of base pairing with a thymine in DNA, it will base pair with a uracil in RNA. T will still go to A, C will base pair with G, G, C, and once again we have an A, does not base pair with a thymine. There is no thymine in RNA. There's a uracil. So now we have our single strand of RNA that can now go and leave the nucleus and attach to a ribosome for translation. And at this point, the DNA strands that have been separated in this transcription bubble can go back together and form our original double helix. And this takes us through transcription of DNA. So hopefully at this point, you've got a good understanding. It's a pretty simple process, especially if we understand replication already. But you should be able to describe the steps of transcription, define the role played by RNA polymerase, and identify the purpose of messenger RNA, acting as that middleman that can go from DNA to the ribosome.